So the focus of today is going to be thinking about single gene expression and how single genes give rise to phenotypes. And usually for students, this centers around Mendel's laws. All right. So there are pros and cons to thinking about it this way, but this is where we're going to start our framing. So the idea here is that heredity, which is really the focus of genetics, is that we can transmit traits from one generation to the next. And there's tons of evidence for this, but it used to be a controversial theory. And this whole idea of being able to study heredity is where genetics comes from. So, genetics specifically is the scientific study of heredity, and we largely blame Gregor Mendel for starting our lovely field, even though he was not the first person, nor was he the most ethical person who started this work. And most students are familiar that Gregor Mendel worked on pea plants and that he was able to trace generations and traits across all of the peas that he mated. And he had this hypothesis that there are alternative versions of genes, which he called alleles, and that these determine the heritable traits that get passed down. And kind of the stereotypical example with his pea plants were things like color. So, one of the things that we want to be comfortable with is the naming of generations and how we're going to talk about them as we go through different steps. So, normally we're going to start with a parental generation. In this case, we have a purple pea and a white pea. That's our parental generation. And when we cross these two together, we get our first filial generation, F1, and all of them are purple. And so Gregor was thinking, what happened to this white trait? Is it just gone? But when he crossed the filial generation, the white trait comes back. And so this suggests to him that this white trait isn't gone. It's still there. The instructions for it are still there. And how does that work? All right. So seeing these traits disappear and then reappear is kind of what got him into this idea of, all right, well, what is determining when they come back? And part of what made peas so helpful is that they have very specific ways of reproducing. And you won't have to explain this on your exam, but just to make it clear how easy it was, this is the basic structure of a pea flower. And so it's very easy to see where the parts of the flower that are female and male are located. So the eggs are going to be located in this structure here, and all the sperm are located in these structures here. And so if you want to control which pea plants are mating with each other, you can remove the structure you don't want. So it wasn't that he let the pea plants just reproduce the way they would naturally, with bees and whatnot. He actually removed the opposite sex materials and then purposefully mated them with each other to guarantee that the paternity, so to speak, was what he wanted. The other nice thing about plants, and particularly peas, is that all of the F1 generation is organized in a nice little packet. That means that it's very easy for him to then take all of the seeds from this generation, plant them, and see what happens. Right? So it's just a very clean model organism to use for this purpose. But it takes a long time, right? You have to grow peas only in cool seasons, so usually spring and fall, and you've got to make sure that the pea seeds are dry enough to be able to be planted. So this took him a very long time. He used the results from these experiments to develop four hypotheses, which we now use for our kind of modern definitions of how alleles and genes function. The first one is that there are alternative versions of genes, and we do call them alleles. And this accounts for the variation in any characteristic trait that we want to follow. Right? So any difference, specific location is going to be called an allele. And this means that many genes often have hundreds of alleles, but we're going to think about only a couple at a time. To give you a very clear example, which is not 100% accurate, but close enough, you can think about alleles like this, right? So these are phenotypes, blue eyes and brown eyes, and they are coming from two different alleles of the same gene. One of these alleles determines if you will have blue eyes, the other allele says that you will have brown. He then hypothesized that for each characteristic or trait, an organism only gets two copies of whatever that gene is, one from each parent. We now call these homozygous or heterozygous depending on whether or not the allele copies are the same. So homo means same or like, and hetero means different. So a homozygous individual will have two of the same alleles. So they might look like this. And a heterozygous individual will have two different alleles. So they might look like this. From there, he hypothesized that the combination of alleles has some kind of interaction. And his was very simple. He only paid attention to phenotypes that had clear distinctions. So if the two alleles of an inherited pair differ, i.e., the individual is heterozygous, then the dominant allele is the one you will see. The recessive allele is covered. From a basic standpoint, we can think about the eyes then like this. The two homozygous individuals contain information specifically about the color we see. Two blue alleles will give blue, and two brown alleles will give brown. If brown is dominant to blue, then brown will show up in the heterozygous individual. So these terms, dominant and recessive, were then applied to how the alleles behaved. We now know it's not as simple as this. In fact, having heterozygosity at this specific allele for brown or blue does not necessarily confer brown. There are blue eye individuals like this and green eye individuals like this too. But for understanding the principles behind what Mendel was talking about, this is generally true. For traits that follow this type of pattern, though, being dominant or recessive has nothing to do with how common the trait is. And this is something that students sometimes struggle with. For example, if we're looking at the four different outcomes here, dominant traits versus recessive traits and their frequency, freckles is a dominant trait and is less common than not having freckles. Being albino is a recessive trait that is less common than having normal pigmentation. So in this case, the recessive trait for freckles, i.e. not having them, is more common. And in this case, the dominant trait of having pigmentation is more common. So don't get into the trap of dominant is more common, because that's not generally true. In fact, many mutations that confer disease are dominant as well and are extremely rare. The other thing to understand about dominance, which Mendel didn't discuss, is that dominance is relative. This happens because there are multiple alleles in the same location. For example, in these coat colors, black, chinchilla, Himalayan, and albino, these are all alleles that occur at the same gene. That means that they all are going to have different dominance patterns. In this case, the black allele is dominant to all of the others. An individual who has this will be black. But an individual who looks like this will be chinchilla. And an individual who looks like this will be Himalayan. Each of the different alleles is dominant and recessive only in a pair. In this situation, the chinchilla allele is recessive. But in this situation, the chinchilla allele is dominant. Whenever we're discussing recessive versus dominant, we are only doing so within the pair of alleles we are currently discussing. So please keep in mind that just because something is dominant sometimes doesn't mean it's dominant all the time. The last of Mendel's hypotheses was that a sperm or egg carry the alleles from each of the intended characters. This is because we know the law of segregation places chromosomes, half as many, into each of the eggs and sperm cells. This allows one copy to come from each parent. The law of segregation is not the same as independent assortment, but it is related. So let's think about this idea. Take the experiment a little further. We started with the true breeding P generation, parental generations of purple flowers and white flowers, and we forced them to reproduce together in our experiment, creating an F1 or filial 1 generation where all of the plants have purple flowers. When we then cross the purple flowers together, the white comes back. When this happens in a standard dominant recessive relationship, we expect to get a consistent outcome. In this case, 3/4 of the plants will be purple and 1/4 of the plants will be white. In order for this to be possible, every cell has to have two copies, as Mendel suggested. These chromosomes are homologous chromosomes. Remember that homologous chromosomes contain the same information from the standpoint of they contain all the same genes and usually in the same orientation. But this information on the gene, the allele it actually has, can be different. This homologous chromosome contains three genes that we've marked, P, A, and B. In the case of the B gene here, we can see that one of the homologous chromosomes carries the dominant allele, whereas the other carries the recessive allele. We can think about then the genotype for the individual based on which alleles are present on each homologous chromosome. For example, for the P allele, this individual is homozygous dominant. For the A allele, they're homozygous recessive. And for the B allele, they were heterozygous. When we think about sets of alleles that tend to go together, for example, these are all physically located on the same chromosome. The set of alleles that tend to travel together are called a haplogroup. We'll discuss haplogroups in a little bit when we talk about how some of this information is useful for thinking about ancestry. The same information that we can see here with our homologous chromosomes also explains Mendel's outcome. For example, in this experiment, we saw that we got purple. Once we see which phenotype is showing up in the F1, we can assign whether or not it is dominant or recessive. Because we saw purple, this tells us that purple is dominant to white in pea plants. So we can define purple with the capital letter P and white with the recessive letter P. Once we do that, we can assign genotypes. But sometimes we need more information. For example, let's say we saw this plant outside of our experiment. What genotype would this plant have? The genotype is the combination of alleles that the plant possesses. Given that the dominant allele is big P and the recessive allele is little p, what genotypes or combinations are possible? The assumption here is that the flower must have at least one copy of the dominant P, but we do not actually know just by looking at it what the second allele is. It's a good idea to say, under this circumstance, that it's either P dash, or say that it could be this or this. So because a phenotype is the appearance of a trait based on genotype, both of these genotypes have the same phenotype. So it is impossible to know which one this plant is without more information. The same is not true for the white plant. What genotype does this white flower have? In this case, we know already that white is recessive. This means that if we assign recessive genotype, it must be homozygous recessive for this trait. So when looking at individuals, if the dominant trait is apparent, and you do not know anything else, you cannot assume that they are homozygous dominant. But if you're looking at a recessive individual, you can assume that they are homozygous recessive. There are a couple of things that you will be able to tell in your questions. For example, when we set up this experiment in the beginning, I mentioned that these plants are true breeding. True breeding means that if they were to reproduce with themselves, they would produce offspring that are identical. This tells you that they are homozygous for the trait. We know already that the white plants must be recessive, but we know that these are homozygous dominant because we were told they were. So these, we need that information, but these ones, we don't. Once we have this, we can then see that all of the F1 will be heterozygous. This explains why all of them are purple, because the purple trait is dominant to the white trait. It also explains why the result should be 3-to-1. Most students are familiar with the idea of a Punnett square. Punnett squares are ways to organize information. In the Punnett square, information from one parent is placed on the top. These are the alleles that the parent could possibly give, in this case, the sperm. And alleles from the other parent, the egg, are placed along the other axis. We can then populate across to fill the gaps and determine what the genotypes could be for each of the empty squares. This tells us that there should be 3 individuals that have at least one copy of the dominant P allele and thus will be purple, and one copy of the white